Hello, everyone, and welcome to this screencast on our introduction to blood. We're going to be studying human blood here. Remember, this is chapter 17, so we're kind of going backwards. I like to cover the heart and the blood vessels first, and then come back and cover the blood. So let's go ahead and get started. First of all, let's take a look at some introductory information. Um, I get a lot of questions every time I'm talking about human blood, and so I kind of put these answers to questions up here on the first slide, and maybe we'll answer some questions that you have based on the blood. So the blood consists of a suspension of special cells in a liquid, and that liquid is referred to as plasma. In adults, the blood is about 1/12 of the weight of the body, and that corresponds to somewhere between 5 and 6 liters because, again, different body sizes, different volumes of blood. So we have 5 to 6 liters, we got that range. The blood actually consists of more than just plasma, though. Blood consists of about 55% plasma and about 45% by volume cells. And these cells we oftentimes refer to as formed elements because, technically speaking, one of the formed elements we're talking about are not true cells. So we'll take a look at that as we move along. Okay, what's the function of the blood? First, the blood transports dissolved gases. We'll talk about more whenever we get into the respiratory system. These gases are oxygen and carbon dioxide, which are necessary. Oxygen is necessary for cellular respiration. Carbon dioxide is a byproduct of cellular respiration. Carbon dioxide needs to be removed. Obviously, oxygen is necessary to drive the process. In addition to these dissolved gases, there are also metabolic waste products produced by the cells, nutrients that need to be provided to the cells, hormones that control body function, and various enzymes that we find transported through the blood as well. The blood also aids in thermoregulation, as we have talked about uh, in other PowerPoints, particularly whenever we were going over the muscular system. It also helps to control pH, and we'll talk more about that actually whenever we get into the respiratory system. The blood also helps to remove toxins from the body. As the blood passes through the liver processes out many of these toxins. And it also helps to regulate electrolytes, the concentration of electrolytes in the blood. These are positively or negatively charged ions like sodium ions, chloride ions, potassium ions, so on and so forth. And again, most of this we're going to be talking about whenever we get into the respiratory system because the blood works very closely with the respiratory system. I should say the cardiovascular system works very closely with the respiratory system to control many of these functions. Okay, so two components of the blood, the plasma and the formed elements. Let's take a closer look at the plasma first. Plasma is a slightly alkaline fluid. What does that mean? It means it has a slight pH slightly above 7. A good pH is usually around 7.35. And it has a typical yellowish color because of the high concentration of proteins that are in it. Plasma, however, consists of about 90% water and only about 10% dry matter, and that dry matter is composed of various proteins that are dissolved in the blood, in the plasma, glucose, mineral ions, hormones produced by various cells released into the blood, and of course carbon dioxide. And there are some others as well. We could talk about the other respiratory gas, oxygen, that's also found in the blood. But that's good for now. We got some good basic materials that make up the dry matter. All right, next up, there are many different proteins that are dissolved in the plasma of the blood, but we want to focus here on three important ones that we'll be talking about not only here in our PowerPoint on the blood, but will become important in other body systems we talk about as well. One of them is albumins. Albumins are proteins that are very important in creating osmotic pressure that's necessary for capillary exchange that we talked about in the cardiovascular system. And albumins are also important because they can kind of piggyback other molecules in the blood to transport them from one place to another. So we'll be seeing albumins again in the future. Next up, we have immunoglobulins. Immunoglobulins are also referred to as antibodies. They're called immunoglobulins because these proteins are globular proteins that participate in the immune system, in the immune response to infection. So we'll be taking a look at antibodies, immunoglobulins, whenever we move into our next PowerPoints, and we're talking about the immune system. And then finally, another important plasma protein. And remember, this is not the end-all, be-all of the plasma proteins, but these are the, we might say, the big three. And the third one is fibrinogen, which is very important in blood coagulation or blood clotting. So we're going to see fibrinogen, and we'll actually talk more about fibrinogen in this PowerPoint. Okay, moving ahead, the other component of the blood is the formed elements. Three important formed elements we have are the erythrocytes, also known as red blood cells, leukocytes, known as white blood cells, and thrombocytes or platelets. So let's take a look at each of the formed elements individually here. Actually, before we get into that, just to give you a little bit of background on these formed elements, where do they come from? How do they get into the blood? Well, the formed elements are actually produced in the red bone marrow. Most of the bones of children produce these formed elements because we find red bone marrow in most of the bones of children because they're growing very rapidly. They need to add blood to their bodies very quickly. And so we have a lot of red bone marrow in the bones of children to help with the formation of the formed elements. However, in adults, we get into the maintenance stage of our life cycle, and so we're not adding tremendous volume to our bodies. We're not growing very rapidly. We're not in that growth phase, and so some of the bones of the body begin to convert red bone marrow over to yellow bone marrow and become more important for fat storage. So in adults, we don't find red bone marrow in as many of the bones, but we do find red bone marrow in flat bones and in the end of the long bones in adults, the long bones like the femur, the humerus, the fibula, the radius and ulna, so on and so forth. You know, those ends, the epiphyses, where we have a lot of spongy bone, trabecular bone, if you will, and in the pores of that spongy or trabecular bone is where we find a lot of red bone marrow, and so that's where we're going to find formed element formation. Okay, in the red bone marrow, this just gives us kind of a hierarchy on this slide. And so we're taking a look here, let's say we're looking at the femur, and here's the distal epiphysis of the femur, and we can see the porous, spongy or trabecular bone there. And within that spongy or trabecular bone, we're going to find a lot of what we call multipotent stem cells. Stem cells are cells in the body that have the ability to differentiate or change into other more specialized cell types, but within a certain realm. So we're going to find some multipotent stem cells here in the red bone marrow of, let's say, these, for example, this long bone, the distal epiphysis of the femur. Now, these multipotent stem cells themselves can undergo differentiation. They can change to become one of several different types of stem cells. They can differentiate or change to become erythroblasts, which are stem cells that give rise to erythrocytes, the red blood cells. Various stem cells for the white blood cells, the leukocytes. There are lymphoblasts that can go on and differentiate to form the lymphocytes, monoblasts to form monocytes, and myeloblasts, which can give rise to three types of red blood cells called neutrophils, basophils, and eosinophils. The megakaryoblast then are multipotent, or rather they are stem cells formed from the multipotent stem cells that can give rise to the platelets of the body or the blood. So just a little bit of hierarchy that you can maybe understand where these things are coming from. Okay, with that in mind, let's begin to take a look at each of the different formed elements more specifically. We're going to begin with the erythrocytes. Remember, those are the red blood cells. Structurally, they have a kind of a donut shape to them, and they're biconcave. Now, they're not like regular donuts that have holes in the middle of them, but whenever we look at them under the microscope, we can see that they are concave or dented in the middle. And this kind of gives a shadowing effect that makes it look like they may have holes in them, but they don't. They're just really thin in the middle and thicker on the edges, and that kind of gives them that donut shape. When we take a look at cells, we have the three main parts of a cell. We have the plasma membrane or the cell membrane. We have the cytoplasm and the nucleus. Well, whenever we look at erythrocytes, one of the outstanding features that we see right off the bat is that erythrocytes have no nucleus in them. The nucleus just takes up space where they'd be able to perform their function. So the nucleus is removed from the erythrocytes as they mature. And the reason for that has to do with their function. Their function is to provide oxygen to the tissues and recover carbon dioxide from the tissues. And the component of the red blood cells that transports oxygen is hemoglobin. And so the more hemoglobin we have in cells, the more oxygen that can be transported. So eliminate the nucleus, leave more room for more hemoglobin, therefore we have more room for oxygen to be transported by the red blood cells. That's the reason why the nucleus is absent. But because they don't have a nucleus, they're not going to be able to repair themselves, they're not going to be able to regenerate parts, make new proteins, and as a result, they're going to have a lifespan of only about 120 days, after which the erythrocytes will be transported primarily to the spleen and liver, where they'll be broken down. The hemoglobin will be broken down, and the cells will be eliminated from the body, and new cells will be produced to replace them. Erythrocyte concentration, these are the most numerous of the formed elements in the blood, and they make up about 4 to 6 million erythrocytes per cubic milliliter of blood. So whenever we take a look at that, how much is a cubic milliliter? Well, that's about a teaspoonful of blood. So what we're saying is that you have about anywhere between 4 and 6 million red blood cells in every teaspoonful of blood that you have. That's a lot of red blood cells. Okay, our second formed element are the white blood cells or the leukocytes, and these are important, and these are cells that are responsible for defense. They're part of the immune system that we're going to be talking about next week. When we take a look at leukocytes, though, we divide them into two categories. One category is what we call granulocytes. Cytes actually means cells, and granulo means granule. So these would be literally granule-containing cells called the granulocytes. So granulocytes do have granules present in the cytoplasm of the cells, and that's where they get their name from. There are three important granulocytes that make up the leukocytes. These are neutrophils, eosinophils, and basophils. And we'll take a look at each one of these individually. But for now, the other category or type of leukocyte is called an agranulocyte. So when we put this A as a prefix out in front of granulo, we get without. So we have literally, the name tells us, these are cells without granules. So there's no granules present in the cytoplasm of these cells. There are two types of agranulocytes. They include the lymphocytes and the monocytes. So whenever we take a look at leukocytes, we classify them as either granulocytes or agranulocytes. The granulocytes are neutrophils, eosinophils, and basophils. The agranulocytes are lymphocytes and monocytes. So let's take a look at each one of these. Let's begin with the granulocytes, and our first granulocyte is the neutrophil. Now, white blood cells, unlike red blood cells, do have a nucleus present within them. However, the nucleus is a little structurally different from one type to another. So when we take a look at neutrophils, the nucleus can be divided in anywhere from 2 to 5 lobes, and those lobes are connected by a fine strand or a filament. So if we take a look at our artist's conception over here, here we can see 1, 2, 3, 4, and just a small one here, and lobes, and we can see the fine strands that are kind of connecting them together. So we're going to have a nucleus that's a lobed nucleus and has anywhere from 2 to 5 lobes. The granules, these are granulocytes, so they are going to have granules present in the cytoplasm. The granules tend to be small and faintly pink colored. So we can see the artist's conception here. We can see the faint pink granules that are present in the cytoplasm of neutrophils. Why do we have neutrophils? Their function is to phagocytize or engulf and remove bacteria. So whenever we take a look at neutrophils, we find that they're going to be present in the pus of wounds. And so one of the things that we see is that neutrophils are kind of frontline defenders. Anytime you get an infection, or maybe whenever you have a wound and you have debris laying around, maybe dead cells, maybe structures that are kind of left over from the wound, maybe even bacteria trying to get in. We're going to find these neutrophils there. They're going to engulf and remove them. But whenever they do, the neutrophils themselves are going to die at the site where they're engulfing and removing, and they accumulate at that site. And when these white blood cells accumulate at that site, that's what we refer to as pus. Concentration-wise, they make up about 50 to 70% of the leukocyte population, not of the total formed element population, but of the leukocyte population. They are frontline defenders, and so we're going to have a very high concentration of them. About 50 to 70% of the leukocyte population should be neutrophils. Next up, we have eosinophils, and we take a look at the we'll do the same here, and we'll look at the nucleus. The nucleus, however, in eosinophils is generally bilobed, and it's usually pretty easily visible. So you can see it pretty clearly. So here's our artist's conception. We can see the 2 lobes of the nucleus, and we can see granules here. Now, these granules, whenever we take a look at them, are pretty abundant in the cytoplasm, but they have a characteristic kind of pinkish orange coloration. And they're going to be more abundant in the cytoplasm than what the granules are in neutrophils. And that's one of the ways we can tell the difference, because, you know, neutrophils can have between 2 and 5 lobes. So if you have 2 lobes, how do you know if it's an eosinophil and not a neutrophil? We're going to look at the granules. We're going to see a more densely populated amount of granules in the cytoplasm, and they'll kind of be a pinkish orange coloration as opposed to that faint pink coloration that we would see in neutrophils. The function of eosinophils, they attack parasites and phagocytize antigen-antibody complexes. So basically what we're going to see is that whenever we get a parasitic infection, neutrophils are going to be called on to attack the parasite, break the parasite down, and phagocytize that parasite to remove it, and also phagocytize antigen-antibody complexes that we'll be talking about in the immune system. So whenever we do get, just to give you a prelude of that, whenever we do get an infection, let's say a bacterial infection, antibodies are going to be released from B cells. Those antibodies are going to bind let's say, bacteria up. And whenever they bind bacteria, you can get a whole kind of a conglomeration or complex of bacteria that are there, and they are neutralized by the antibodies, and then they can be removed by the eosinophils. It's a little bit hard to explain it. You'll need a little more detail, and we'll get into it in more detail whenever we get into the immune system itself. So the concentration of eosinophils not nearly as high as neutrophils. The concentration is only about 2 to 4% of the leukocyte population. So if you're doing a blood smear and you're looking for various white blood cells, neutrophils are going to be the easiest to find. Eosinophils, only 2 to 4% of the population, they're going to be a little more difficult to find. That takes us to the basophils. When we take a look at the nucleus of basophils, it can be bi or trilobed. We can see here in this artist conception a bilobed nucleus. The big thing though here is that nucleus can be very difficult to see due to the high concentration of granules. And this artist conception really doesn't do it justice. The granules are very, very densely packed in the cytoplasm, and the nucleus can be almost completely obscured and very difficult to pick out whenever we take a look at basophils. These granules tend to be dark purple in color, and as I mentioned, very abundant, very densely packed in the cytoplasm. The function of basophils, their job is to secrete anticoagulant and vasodilatory substances. So anticoagulant, things that stop blood clotting, and vasodilatory substances, these are chemicals that will cause blood vessels where an infection is going on to dilate or to open up to allow greater volumes of blood, bringing in neutrophils, eosinophils, and other immune system factors to help fight off that infection. So you don't need a whole lot of basophils. You just need some there to produce these chemicals to signal anticoagulation, to stop coagulation, and to produce the chemicals that dilate blood vessels. So they're the rarest in terms of concentration. They comprise less than 1% of the leukocyte population. If you're doing a blood smear and looking for basophils, you can spend a lot of time looking around trying to find a single basophil. Okay, moving on to the agranulocytes now. Remember, we had lymphocytes and monocytes. The first agranulocyte we take a look at is the lymphocyte. The lymphocyte is fairly easy to identify whenever you see it in a microscope slide. That's because the nucleus of a lymphocyte is very large and round, and as we can see here in this artist conception, it's going to take up almost all of the cytoplasm of the cell. As far as granules are concerned, this is not applicable. These are agranulocytes. The lymphocytes, however, their function, they're the main components of the immune system. Lymphocytes produce antibodies, or at least a specific type of lymphocyte produces antibodies, which are very important in the immune response. So we're going to be up in concentration on these lymphocytes a little bit because of their importance. Their concentration is going to be anywhere from 20 to 40% of the total leukocyte population. The monocytes, on the other hand, also have a very large nucleus. However, we can identify them easily from the lymphocytes due to the fact that their nucleus is kind of horseshoe shaped, or what we call reniform. You can see this large indentation in the nucleus. Still takes up most of the cytoplasm, but it has that characteristic horseshoe shape to it. Granules are not present. Again, these are agranulocytes. The function of monocytes is to migrate into the connective tissues where an infection may be going on. And they can fuse with one another to become large phagocytic cells we call macrophages. And therefore, they can do like the neutrophils, they can phagocytize microorganisms. Concentration of monocytes is about 3 to 8% of the circulating leukocyte population. Okay, so we then ask ourselves the question, how can we remember what's the highest amount, what's the lowest amount? Well, here's an easy mnemonic to help us remember the various concentrations of the leukocytes. It's never let monkeys eat bananas. It's kind of a goofy little mnemonic, but it's an easy one, I think, to remember that you can use. And we take the first letter of each word to represent the various concentrations, with the first word never for neutrophils. So running from left to right, we're going to go from highest concentration to lowest concentration. So never N neutrophils, they're about 50 to 70% of the leukocyte population. Let lymphocytes about 20 to 40% they should be. Monocytes should be in a range of about 3 to 8%. So E or M for monkeys. E in eosinophils about 2 to 4% of the total population, and B, bananas, for basophils should be less than 1% of the population. If we have concentrations outside these ranges, that can help with diagnosis of various disease states. Okay. Finally, then, we have the thrombocytes, the third important formed element. Thrombocytes are also known as platelets, and this is the reason why we have the name formed elements instead of plasma and cells. We have plasma and formed elements because platelets are not true cells. They're formed in the bone marrow from big leukocytes, as we saw in our hierarchy, called megakaryocytes. Interestingly, platelets have sticky surfaces. Only whenever they become damaged, they become sticky. And as a result, they have an important function in blood clotting to help accomplish what we call hemostasis. Hemostasis, the stoppage of bleeding. So the platelets can stick to one another and also stick to the tissue of the injured site. And as a result, they can kind of form a plug that can temporarily stop the flow of blood whenever injury to a blood vessel occurs. So whenever we take a look over here, this is an actual micrograph, and we can see red blood cells here, their characteristic donut shape, and then we can see these clusters of kind of purplish cells. These are the platelets. Platelets also, in addition to having those sticky surfaces, can release, whenever they become damaged, they can release clotting factors. And these clotting factors are substances that can trigger the blood clotting process, trigger the process of stopping the bleeding and healing the blood vessel over eventually. Speaking of the clotting process, that's the next topic I'd like to discuss with you, is how blood clotting occurs. It occurs along a cascade of events, or kind of like a domino effect. And so we're going to take a look at that, at how this blood clotting occurs along this domino effect. Now, in order for blood clotting to occur, we talked about the factors that were released by the platelets. There are, however, more clotting factors that are present actually just even in the plasma of the blood. These clotting factors are substances in the plasma that are involved in the blood clotting process. Most of them are proteins, either enzymes or enzyme precursors. What's the difference between an enzyme and an enzyme precursor? Enzymes are active. They can do their job that we typically think of with enzymes. Enzyme precursors are enzymes that are in an inactive state. So they have the ability to act upon a particular substrate, but they're not in a form that they can act on that substrate. They're missing something and they can't act on the substrate, so they're precursors. So these clotting factors then would also include coenzymes and cofactors. These are substances that would help the enzyme precursors to become active and to act on the substrates they're designed for. When we take a look at these clotting factors, we give them numbers, Aramaic numbers 1 through 13, or Roman numerals, Roman numeral 1 through Roman numeral 13. But they also have names as well, although we're primarily going to focus on the numbered uh, identifications for these clotting factors. So once the tissue in a blood vessel wall is injured, an initial blood clotting factor has to be activated. Once that initial blood clotting factor is activated, there is then a cascade, or as I mentioned, a domino effect, which with each factor activating the next. So it's kind of like once you topple that first domino, that domino causes another to fall, causes another to fall, causes another to fall. In the case of the blood clotting cascade, once you activate that first factor leads to the formation of another factor, which leads to the formation of another factor, and so on until eventually the blood clot is formed and we're well underway to healing the blood vessel. So here are the blood clotting factors. And I put this up. It's not. I don't put this up for you to memorize all these names. That's beyond the purview of what we're trying to do here right now. But I do want you to see. We got the factors in Roman numerals 1 through 13. Each factor does have a name, so we're showing the name. Factor 1 is fibrinogen. And we also have the pathway. We're going to be talking about extrinsic and intrinsic pathways involved in blood clotting. If we see both here, it means that fibrinogen, in this case, is involved in both the extrinsic and the intrinsic pathway. Whereas if we go down to number 3 here, thromboplastin, also known as tissue factor, is part of the extrinsic pathway, not part of the intrinsic pathway, so it's listed as extrinsic only. Whereas if we come down here to Roman numeral 9, it's called Christmas factor, and it's involved in intrinsic only. So the chart shows which the Roman numeral name, the actual name, and then the pathway that it's involved in. Okay, so we mentioned there are two pathways involved in the blood clotting cascade. One of them called extrinsic and the other one called intrinsic. Let's take a look at these two pathways. The first one we're going to look at is the extrinsic pathway. The extrinsic pathway is activated when blood comes in contact with tissue factor expressed following the rupture of a blood vessel. So, what we find is that, let's say, for example, we do something like twist our ankle, and as a result of twisting the ankle, blood vessels rupture. When the blood vessel ruptures, what can happen is the blood can flow out, and it can then come in contact with tissue factor. Now, if we go back, it can come in contact with what? Thromboplastin factor number 3. This is extrinsic, right? So this factor is present in tissue immediately surrounding the blood vessel, or we didn't talk about this when we went over the structure of the arteries and veins, but we'll talk about it now. Surrounding the endothelial tissue, between the endothelium and the internal elastic membrane, there is a very tiny amount, a very thin layer of connective tissue. And the cells of that connective tissue have, as an integral protein in their plasma membrane, tissue factor or thromboplastin. So when the blood flows through the lumen of the blood vessel, that thin layer of connective tissue that contains the thromboplastin, the tissue factor, is separated and kept apart from the blood, so the platelets never really come in, or the blood never really comes in contact with it. But when the blood vessel ruptures, these integral proteins, this tissue factor, can become exposed, and the blood can now come in contact with that tissue factor. Okay, so this would set off the extrinsic pathway or domino effect. The extrinsic pathway is also activated by external trauma that causes the blood to escape from the vascular system. So extrinsic pathway can be activated when the blood vessel ruptures and the blood comes in contact with that thin layer of connective tissue that has the tissue factor in it, or as the blood flows out into the surrounding tissue and becomes exposed to that surrounding tissue and the tissue factor that way. The intrinsic pathway, on our hand, is activated by trauma within the vascular system itself. So it's activated when platelets adhere to collagen, which is exposed on the endothelial cell surfaces. So over the course of time, what can happen is cells and other structures can rub against the endothelial cells and cause exposure of collagen hanging down into the lumen of the blood vessel. And what can happen then is platelets can start to adhere to that collagen that now is exposed on those endothelial cell surfaces. And when the platelets adhere, that can set off this intrinsic pathway. Now, this can lead to the formation of clots, let's say like a deep vein thrombosis, or plaque buildup within the blood vessels as well. Because remember, once those platelets adhere to the collagen, they become sticky themselves and other things can stick to them. And this can result in the formation of clots inside the blood vessel lumen. The intrinsic pathway is slower than the extrinsic pathway, but it is more important because of the formation of those clots. It may be activated also by the contact of the blood vessel wall by lipoprotein particles. So lipoproteins or cholesterol can come in contact with the vessel wall and set off the intrinsic pathway as well. Now, in this PowerPoint, I'm not going to go over the specifics of the blood clotting cascade, but I'm going to refer you to this YouTube video I'd like you to watch. It is important to watch it and to learn. I can't explain the blood clotting cascade any simpler than it's explained in this video, so I'll leave it to the video for you to watch, but I want you to go see the video, Blood Coagulation Clotting Cascade, which I've posted on Canvas to make a quick link to it for you under videos and animations. So please make sure you go to do that. I'm going to pause our blood PowerPoint here, our lecture on human blood here, to allow you to go take time and watch that blood clotting cascade video. And then we'll come back in our second lecture and we'll talk about some things related to what you're going to learn as you go through that blood clotting cascade video. So we'll stop there for today, and in part 2, we'll pick up from there and move to the end of our discussions. Have a good day, everybody.